Welcome and thank you for joining us for today's webinar, where we will explore the chemistry of organosilane reductions for use in pharmaceutical synthesis. My name is Gabby Lockwood, Marketing and Communications Specialist with Gelest, headquartered in Morrisville, Pennsylvania, United States. Since its founding in 1991, Gelest has been and continues to be recognized as a world leader in the development and manufacture of organosilicon and metal organic materials, serving advanced technology markets through a customer-driven approach. With an offering of materials that enable innovation, Gelest provides focused technical development and application support from inception to commercialization for pharmaceutical synthesis, optical materials, semiconductors, diagnostics and separation science, specialty polymeric materials, functional microparticles, and cosmetic materials. In an effort to share this expertise with you, our esteemed colleagues, we are pleased to present the first in our series of educational webinars. I'm honored to introduce Dr. Jerry Larson, who will speak on today's topic of organosilane reductions. Jerry is a PhD chemist with a long involvement with organosilicon chemistry. He is currently a Senior Research Fellow at Gelest, following several years as Vice President for Research Products. Formerly, he was Director of Research at Degussa Corporation and Professor of Chemistry at the University of Puerto Rico. His responsibilities include the development of Gelest synthetic literature, presenting lectures at various technical venues within the pharmaceutical and fine chemical industries, and the development of new products and processes. During the webinar, all attendees will be on mute. If you have questions, please submit them anytime throughout the webinar via the Submit Question option in the chat window provided, or preferably via an email message to Dr. Larson. Questions will be addressed at the conclusion of the webinar through an email response as soon as possible. All Q&A will be distributed to all attendees. So please sit back, relax, and enjoy learning about reductive transformations for organic synthesis.
Hello, fellow chemists. I'm Jerry Larson, Senior Research Fellow at Gelest Incorporated. At Gelest, we make a wide range of organosilicon compounds. The majority of these go into industrial applications, as well as many synthetic organic applications. We at Gelest make a range of metal organic compounds as well. Today, I'm going to share with you a line of products that we use for reductions, namely organosilane reductions. Although there's a variety of organosilanes, today's topic is really going to cover one of those, namely tetramethyl disiloxane, otherwise known as TMDS. TMDS is a small molecule, and it has two hydrogens available for the reduction. It has a boiling point of 70 degrees at atmospheric pressure, and it has a flash point of 10 degrees. So there are some handling issues to be precautioned with in using it, but it is relatively easy to handle and very reactive. General considerations involving organosilane reductions include the SiH, of course, is the source of the hydride. That hydride and the SiH bond is relatively weakly hydridic. That means that it can be very selective in its reactions because the reactivity is somewhat lowered. And therefore, you also need acid or sometimes fluoride catalysis to get the silane reductions to occur. So that means that carbocationic intermediates are commonly employed and involved. Organosilanes are easily handled. Organosilanes are thermally and hydrolytically stable as long as the pH is near neutrality. Organosilanes can be sterically and electronically modified to fit specific needs if need be. Here's a list of some of the more commonly employed organosilane reducing agents. So you have triethylsilane is very popular. It's used in the synthesis of Tamiflu, for example. Phenyl dimethylsilane is also used in a number of organosilane reductions. Phenylsilane has some special applications and so forth. The PMHS is a polymeric organosilane with silicon hydrogen bonds. It is the least expensive of the silane reducing agents. It is easy to handle and it can provide facilitated workups at the end of the reaction. The TMDS shown in the middle of the slide is what the topic will be for today, as I mentioned already. Some of the costing aspects for organosilane reducing agents is shown on this slide. On the left you see the silane reducing agent, a little bit about their properties, the silicon byproduct that you would find after the reduction has taken place. So this is something you would have to consider during the workup of an isolation of your product. And then the final column is that which is giving you the cost per mole of hydride, assuming we're making 1,000 kilograms of the silane. And what we see here, these numbers are an idea of the relative cost. The triethylsilane coming in at about $10 per mole of hydride, and the PMHS on the other extreme at 90 cents per mole of hydride, so 1/10 roughly the value. TMDS, the topic for today, at about $3 per mole of hydride if you're making a metric ton or more of the reagent. So what I'd like to do now is to share with you in the next several images some of the reductions that TMDS is capable of. For example, amides are reduced very nicely with TMDS and triiron dodecacarbonyl catalysis to the aminomethyl derivative. And you see on this image that the yields are very high, and one sees in this example 6 examples where an ester is tolerated, a bromide is tolerated, and a number of other groups have been tolerated. Both aromatic amides and aliphatic amides are reduced. This is a very, very good alternative to the typical conditions for this reduction, which involves diborane or borane THF complex, a very difficult material to handle. The next image, we'll see a different approach to the amide reduction, and that is with chloroplatinic acid instead of the triiron dodecacarbonyl catalysis. The yields are excellent, and the conditions are rather mild at 4 or 5 hours at 75 degrees. And here we see toleration of a nitro group, a cyano group, and again an ester. The amide is reduced selectively in the presence of these other functionalities. It has also been shown to be possible to reduce an amide, a very difficult group to reduce, to an aldehyde, which is typically a very easily reduced group. So this is interesting. On the left you have the amide, TMDS, very inexpensive catalyst, the tippet or titanium isopropoxide, and you get the aldehyde as the final product in very high yields. Tertiary amides are reduced very nicely. Secondary amides, as shown in the third example, reduced very cleanly to, in this case, benzaldehyde. And in the one example of a primary amide that was looked at gave a very, very poor yield of the corresponding aldehyde. So tertiary amides, secondary amides reduced to the aldehyde very nicely. Interesting in that a difficult to reduce amide group can be reduced to an aldehyde, which is a very easily reduced group without further reduction. In those examples where the amide to be reduced has alpha protons available, iridium catalysis will carry out the transformation of that amide to a corresponding enamine, so loss of water. Tertiary amides work very well in this process. TMDS is the best silane reducing agent for it. However, PMHS works equally well. If the product can be isolated from the PMHS, this would be a good alternative. Anilines are present in a number of biologically active compounds. One common route for the synthesis of anilines is the reduction of a nitro group. The next set of images will demonstrate how TMDS can be used in this highly useful reduction. And the examples shown in the image before you on the iron carbonyl catalyzed TMDS reduction of paranitro dimethyl benzamide gives you the paramino dimethyl benzamide. So the reduction of the nitro group is preferred over that of the amide, another unusual transformation. However, if the conditions are such that the chloroplatinic acid is used as the catalyst, reduction of the amide takes place, the nitro group is untouched. TMDS will reduce various aromatic nitro groups in high yields without any problem. The selective reduction of aromatic nitro groups to the corresponding anilines has also been carried out by gold nanoparticles incorporated into iron oxide. The equation for the formation of these nanoparticles is shown on the image before you. Sodium hydroxide reduction of tetrachloroauric acid to the gold nanoparticle. These catalyze in very low catalytic concentration the aromatic nitro to the aniline. And you can see by the number of examples here, the yields are very, very high. And most importantly, one can tolerate hydroxyl groups, benzyl alcohol even. One can tolerate a ketone group, an ester group, an amide group, a nitrile group, and so forth. Very, very selective reduction of the nitro to the aniline. Very important transformation. So in summary, one has conditions available to reduce an amide in the presence of a nitro group, as shown with the chloroplatinic acid catalyzed reactions. One can reduce the nitro group in the presence of the amide, on the other hand, in the presence of the triiron dodecacarbonyl catalysis. And one can reduce the nitro group in the presence of the amide with the gold nanoparticles in the iron oxide medium. Now the next set of images I'd like to show you involves the reduction of nitriles, again, using the TMDS organosilane reducing agent. One very attractive transformation is that of the conversion of the nitrile to an aldehyde group. This is carried out with triisopropyl vanadium oxide in an equivalent amount. The nitrile is converted in high yields and decent yields to the corresponding aldehyde. As shown in the examples here, the nitro group is tolerated without reduction to the aniline. In the next set of images, the reduction of a nitrile to the amine, the primary amine. The example shown here, the amine was isolated as the hydrochloride salt, and instead of the free amine, that proved to be easier to do. TMDS, the reducing agent again, tippet is the catalyst. The amines, hydrochlorides, are isolated in very, very high yields. This reduction is typically carried out with hydrogen and Raney cobalt or Raney nickel as the catalyst. This offers a very nice alternative to that chemistry. Dinitriles were shown to be reduced, but not to the diamines, but rather to azocyclic species. Again, isolated as the hydrochloride salts, these reductions were shown to produce 4-membered rings, 5 and 6 and 7-membered rings, all in quite acceptable yields. On the next set of images, I'd like to share with you the reduction of carboxylic acids to the corresponding alcohol and alkyl iodides. This is typically carried out with lithium aluminum hydride or a similar quite strong reducing agent, but under the conditions using TMDS and indium tribromide as the catalyst in these reactions, the reduction is carried out under very mild conditions, very high yields, the carboxylic acid being formed, transferred to the primary alcohol in very, very good yields. Aliphatic carboxylic acids, aromatic carboxylic acids, all work very well. One can note that the nitro group in this reduction sequence is tolerated, and aliphatic nitriles are reduced to the primary mean, just as are aromatic nitriles. In the next image, we will see what happens with diacids. Appropriate diacids are reduced and subsequently cyclized to the cyclic ethers. This has been shown to occur for the production of 5-membered ring ethers, 6-membered ring ethers, and in one case a 7-membered ring ether as well. Again, the yields are quite good, although some of the yields leave room for improvement. Reductive iodination of a carboxylic acid is also possible. The reduction takes place to the alcohol treatment with iodine, or trimethylsilyl iodide can also be used in this transformation. One ends up with the primary alkyl iodide as the final product. The yields of several products are shown here in the image. You can see that they're in the high 80s, typically to the 90% range. Unsaturated aliphatic carboxylic acids react but give a complex mixture of products, presumably due to the reactive double bonds with the iodine, and so isolation of the desired product is not possible in those instances. It's also been shown that one can use TMDS to reduce the iodides or triphlates to the corresponding alkane. A carbene copper catalyst proved to be best. Cesium fluoride to activate the silane in these reactions is necessary as well. Very good yields of the corresponding alkane from the alkyl iodide are obtained. Iodides as well as triphlates are reduced under these conditions. It is interesting to note that the tosyl group is tolerated under these conditions. The triphlate group is reduced, but not the tosyl group. Also of interest is that the T-butyl dimethylsiloprotected alcohol, the TBS ether, survives these reaction conditions, which is quite interesting in that cesium fluoride is part of the reaction medium. This prompted Lalick and co-workers to do a two-step, one-pot reduction of alcohols to alkanes, wherein the alcohol is converted in the first step by triflic anhydride and 2,6-lutidine to the triflate, which is not isolated but subsequently reduced in the same reaction flask to the alkane. What you see in the table is a variety of functional groups on the benzene ring, an ester, a nitro, a cyano, for example. The yield of the triflate, the yield of the alkane, and then the overall yield. And you can see that the overall yield for this two-step, one-pot reduction of alcohols to alkanes are really quite good. And the bottom part of the image, one can see the tosylate is unreacted. The triflates do react, the tosylates do not. And interestingly enough, the tertiary butyl dimethylsiloxy group remains intact even though cesium fluoride is present in the reaction mixture. And we all know that fluoride goes after silicon very strongly, so it's a bit surprising actually that this TBS ether remains intact under these reaction conditions. The next set of images will demonstrate the reduction of anisols and carboxamides. These are groups that can direct chemistry to an adjacent site. Subsequent to the reaction at that adjacent site, it is often desirable to remove the directing group. While TMDS will reduce an anisole to the aromatic hydrocarbon using nickel 0 as a catalyst and a phosphine ligand. The reaction gives very good yields of the corresponding aromatic hydrocarbon. The methoxy group is gone. It tolerates esters, as can be seen in the image. It also will tolerate an acetal and a diazine. In addition, an oxazolene group has been shown to be tolerated. The next image will show an application of this, and so you see this aerial methyl ether, the nickel zero catalyst, the phosphine ligand, and one gets 62% yield of the corresponding alkane. The benzylic methyl ether remains untouched in this chemistry. The steroidal methyl ether shown in the image is similarly reduced in 62% yield to the corresponding hydride. Carbamates are also very good directing groups for ortholithiation. And so it would be good to have a carbamate, an aerial carbamate, to be able to do the ortholithiation, carry out the chemistry of that new resulting lithium reagent, and then removal of the carbamate so that you can generate new species carbamate free. Just to show the applications and the spectrum of these reductions, there are numerous examples on the image before you. One can tolerate a ketal ethers, and in the one case, a 1-methoxy group is reduced and the other one is not. An ester group is tolerated as well. The yields are all quite good in the high 70s to mid 90%. In the next image, we'll show what can be done here. If you start in the middle of the image, you have this 4-methoxy N-diethyl carbamate. This can direct lithiation with secondary butyllithium and TMEDA, and then that lithium reagent can be trapped. And on the image to the right, this is trapped with triethylchlorosilane to give you the silylated derivative and 71% yield. Then the nickel-catalyzed reduction of the carbamate gives you the triethylsiloanisole as the final product. So we're doing the directed lithiation, we're doing the electrophilic substitution, and then we're removing the directing group, the carbamate. Going to the left in this image, one does the lithiation with secondary lithium again, and then traps with trimethylborate. So you get the boronic acid ester, isolated in this case as the pinacol ester, and 67% yield, and then reduction of the carbamate in the presence of the aerial boronic ester to the metaboronic acid shown to the lower left. So one can see the advantage of the directing group and then removal of that directing group. Another example where we're doing what's called Sine substitution with some cross-coupling. The Sine substitution simply means we are substituting on the adjacent carbon. So we have an indole with a 5-carbamate group attached to do the directing of the lithiation. This directs that lithiation to the 4 position, which is very difficult position in indoles to substitute, so this is potentially very attractive. We put the boronic acid on there as the ester, and then we reduce off the carbamate. That gives us to the 4 boron substituted indole. This boronic acid ester then can be and was subjected to some cross-coupling chemistry just to show its utility. In the one case, reacted with morpholine to give the corresponding 4 amino substituted indole, and in the other case, it was cross-coupled with bromobenzene under palladium catalysis to give the 4-phenyl N-methyl indole as well in good yields. It has also been shown that TMDS can reduce phosphine oxides under catalysis with tippet, otherwise known as titanium isopropoxide. Very important transformation. As you all know, phosphines are very important ligands for a number of catalysts, and palladium in particular, platinum, copper, and others for various reactions, all very important, including cross-coupling. And what we see in the image here is that phosphine oxides are converted very nicely under mild conditions to the corresponding phosphine at very high yields. These are true of even phosphines that contain a phosphorus hydrogen bond, which is normally quite reactive. The phosphine is produced again in high yield. Diphenylphosphine, for example, the bis 4-methoxyphenylphosphine as another example. Azavitic reactions are this is the reaction of an azide with a carbonyl. to form an imine. The one we're showing here is a cyclization. So triphenylphosphine catalyzes that reaction. What one can do is use only a catalytic amount of the triphenylphosphine and the TMDS as a reducing agent. So what we're going to do is reduce in situ the triphenylphosphine oxide that's generated in the azavitic reaction back to the triphenylphosphine to catalyze again the azavitic reaction, and so you get this cyclic turnaround of the appropriate triphenylphosphine reagent. This reaction works very, very well. You get high 90s, low 90s, 90% plus yields of the azavitic product. The next image will show you a number of examples and the corresponding yields for the cyclic azavitic reaction that was illustrated on the previous image. Yields are very, very high. This chemistry was used to produce methaqualone, was used to produce deoxyvacicinone and vacicinone as well, all in very, very high yields. At this point, I would like to turn our attention to some specific examples in the pharma industry where organosilane reductions are extremely useful and highly used. One of these is comprised of a series of compounds that are designed to address the diabetes 2 problem. This is a very serious problem worldwide, especially in America, Europe, and increasingly in Asia. These compounds fall into a class of drugs known as SGLT2 inhibitors. The SGLT is short for sodium glucose cotransport. The drugs collectively are known as gliflozins, and there are similar materials under study that fall into this class. I would like to discuss some of them and where the organosilane reductions play a role. So in the image before you, you see dapagliflozin, which is in the US called Farxiga, and in the Europe called Forxiga. It's a Bristol-Myers Squibb AstraZeneca effort. Recently, FDA approved, I think, in the last 3 months. A very similar structure is canagliflozin, or called Invokana. It's a Janssen product, Janssen, a division of J&J, Johnson & Johnson. It's been approved in the US. A third candidate in this class of anti-diabetes drugs is ipragliflozin. It's produced by Astellas Pharma in Japan, and it's been approved very recently for use in Japan. And you will notice that all of these structures have a glucose sugar that you see on the left side of the molecules, and at the anomeric carbon have an aryl group coming off beta in the beta isomeric position. And so what you need to do is put the hydrogen at the alpha position of this sugar. The next image will show a couple more candidates in this class. One is empagliflozin, a Boehringer Ingelheim, Eli Lilly joint effort. This is in the FDA approval process, should be approved quite soon. That's the expectation. Pfizer has a candidate, erfugliflozin, shown in this image. This one does not have the hydrogen at the anomeric carbon, but in fact, that anomeric carbon is still in a ketal form. Well, where does the reduction come into play in these structures? In 2 places, actually. In the aromatic ring, will eventually be the aromatic substitution at the anomeric carbon. One starts with a Friedel-Crafts acylation reaction. That gives you a diaryl ketone. The silane reduction takes place, or can take place, there are other ways to do that, to convert that carbonyl down to a CH2, a methylene group. And then one can convert, in this case, the aryl bromide to the lithium with a metal halogen exchange reaction. This then is reacted with the lactone, protected, the 4-hydroxyls are protected in this case as trimethylsilyl ethers. The lactone is reacted. This gives you a hemi-ketal at the anomeric position. Now the problem is to do a selective reduction of the hemi-ketal to the ether. This is best carried out with a silane Various silanes could be used, but it turns out that the TMDS is favored in this reduction. So the hemiketal is then reduced to the cyclic ether. Deprotection, removal of the trimethylsilyl groups gives you the final drug, in this case, dapagliflozin. And the next images shows you how this is carried out with canagliflozin. So you see the immediate precursor, the hemiketal, on the left protected as the acetyl ester groups. TMDS and aluminum chloride as the catalyst in this case gives you the ether. The hydroxyl is removed, hydrogen is introduced, and then deprotection gives you the canagliflozin as shown. Another interesting example from the pharmaceutical industry is that of ciprazidone, an antipsychotic drug from Pfizer Pharmaceuticals. Structure is shown on the image before you, and the key intermediate is the corresponding chloroethyl aryl system, which is then substituted by the substituted piperazine. How this chemistry was initially carried out is shown in the image here. So you do a Friedel-Crafts acylation with chloroacetyl chloride. That gets you to the acetophenone derivative shown. Reduction initially was carried out with triethylsilane. The problems here were really the formation of three different products. In addition to the key intermediate shown in red, one had the partial reduction product, that is the benzyl alcohol or the chlorohydrin shown at the top of the image, but also over-reduction to the ethyl group, so the reduction of the chloride as well. This resulted in very difficult purification of the final product to carry on and was deemed undesirable. So this was looked at by the process chemists. The process chemists came up with what's shown in the image as well. You start with the aromatic system. You do the Friedel-Crafts acylation with aluminum chloride and dichloromethane. You get, as an intermediate, not isolated, you get the acetophenone derivative, the same one as up top. And then TMDS, aluminum chloride reduction of this was very, very clean in reduction of the carbonyl to the methylene group, and resulted in much higher yield and much greater purity of this key intermediate and for the formation, ultimately, of ciprazidone. An additional pharmaceutical example of how the process chemists carry out their task is shown in a lactone to lactol reduction in a Janssen Pharmaceutical selective estrogen receptor modulator drug. So the task here is to reduce a lactone to a hemiacetal, as shown. And what the process chemists were given to carry this out was a dichloromethane as the solvent, diisobutyl aluminum hydride as the reducing agent, and then the product. The issues in this process, number one, dibol H or diisobutylalane is pyrophoric. Makes it very, very difficult to handle. Secondly, dichloromethane, being a chlorinated solvent, is undesirable from a number of standpoints, among which are health and environmental concerns. And it required 15 liters of the solvent per mole of the final product. Control of the diisobutylalane stoichiometry turned out to be a problem because residual aluminum salts in the reaction mixture created problems in the subsequent chemistry that had to be carried out. And finally, the generation of waste in this process was something of the order of 60 to 90 kilograms of waste for every kilogram of desired product. This is a lot of waste, a lot of which, of course, contained chlorinated hydrocarbons, dichloromethane, expensive to waste. So the process chemists were charged with the task of solving these problems. And so what they came up with was, as a precatalyst, the difluorotitanosine as the precatalyst. This is actually generated with the silane reducing agent to the actual active catalyst. And they used PMHS in this process. If you recall, the least expensive of the silane reducing agents, of the lactone to the lactol, or the hemiacetal. In comparison, you have to activate the catalyst, so that's a step that needs to be taken place. Hydrogen is evolved during that activation, so there is some hydrogen evolved. Hydrogen is evolved during the desilylation step because the initial product is actually the silylated hemiacetal. You have to then add the tetrabutyl ammonium fluoride to remove the silicon. The yields were about 93%, the purity levels about 70 to 90%. Only 5.5 liters of reaction medium, i.e. toluene in this case, and THF, were needed per mole of the product. That's down from 15.5 liters, so by a third. The waste was generated also about a third of what was initially generated in the first step. And the subsequent steps were much higher yield and higher purity than the initial. So an overall yield is improved, and the overall purity is improved, and also the ease of the subsequent steps and the yields and product purity in those steps is improved. In conclusion, I'd like to show you some of the Gelest publications which serve the needs of the synthetic organic community. I would be remiss not to mention the book that I wrote together with Jim Fry. It's an organic reactions chapter on ionic and organometallic catalyzed organosilane reductions. This book is available from Wiley. In addition, we at Gelest produce a variety of brochures to help the chemists along the way. And we have 3 of these that involve synthetic applications of organosilanes. One of these is silicon-based cross-coupling reagents, as shown here. Another is silicon-based reducing agents. There's considerably more information than today's lecture was allowed to present in terms of time. And finally, we have silicon-based blocking agents. These are, of course, silicon-based protecting groups for synthetic organic applications. Thank you very much for watching our presentation on organosilane reductions. I hope you have gained a greater understanding of and appreciation for the synthetic potential of organosilane reduction. If you have any further questions or need any additional information, please feel free to email us at info@gelest.com or personally email me at jlarson@gelest.com. Thanks again, and the best of success with your chemistry.
Thank you for joining us for the Gelest webinar series. Keep an eye on your email inbox for future invitations to our online seminars. We look forward to sharing the newest and most pertinent information with you. I'm Gabby Lockwood, wishing you the best from all of us here at Gelest, where we will continue enabling your technology.